I'm Patt Morrison. I'm not a Caltech grad, but I guess you could call me a camp follower. I have a lot of Caltech friends, and one of them is your latest Nobel laureate, Dr. Frances Arnold, and I went with Frances to Stockholm when she received her Nobel Prize. So I guess that makes me Nobel adjacent with 1 degree of separation. Now, the original intent of the alumni seminar was to keep alumni in the loop with Caltech's cutting edge research and the faculty who were behind it and how their work is being leveraged in the world today. And you're going to be hearing a lot of that today. The first Caltech alumni seminar was held in 1938, which turned out to be a pretty good year for science. It's the year that Teflon was discovered accidentally. It's the year that nylon was patented and given its first application, which was toothbrushes. Einstein and Leopold Infeld published The Evolution of Physics that year. Alan Turing finished his PhD thesis that year. Otto Hahn and Lisa Meitner discovered nuclear fission, and LSD was developed, 1938. Here on campus in 1938, some of the people conducting those first alumni seminars were names that you may associate more with buildings on campus, like Millikan and Beckman, right behind me there virtually. And they asked questions in this seminar in 1938, questions like, do you know about genes and hormones and enzymes? What do you know about the expanding universe? So we're going to be hearing equally profound questions from speakers who are just as exalted in their fields today and just as significant to us now. So here we go. To start our universe of knowledge expanding is Thomas Rosenbaum, who is Caltech's president, of course, the Sonja and William Davidow Presidential Professor and Professor of Physics. Thank you very much for joining us.
Let me start by thanking you, Pat, for the wonderful history and the generous introduction. Welcome to all of you to the Institute's 83rd Seminar Day, first virtual rendition. Thousands of alumni and members of the greater Caltech community have signed up from across the country and the world to mark this special occasion, a wonderful statement of engagement. Our lives are now suffused with Zoom meetings, teleconferences, online classes, and all kinds of virtual events. But I want to emphasize that we are not a virtual community. We have pulled together to keep the Institute moving forward, even as the whirlwind of the coronavirus pandemic has altered our lives and often increased our burdens. I am proud of how we have supported each other as a community and how our COVID-19 research on campus and at JPL will help the world. Today, we have an extraordinary lineup of presentations and presenters, starting with the incomparable Davidow. Over the course of Seminar Day, we will bounce from virology to AI and machine learning, to planetary exploration, to economics and history. We will look back and we will look forward, but always with the gimlet-eyed view of individuals who believe in truth and data, and who know that asking penetrating questions reveals far more than any answer. As I look at the lineup today, I am reminded of the poet Theodore Roethke's injunction that what we need are more people who specialize in the impossible. On campus and at JPL, we are gearing up for a staged expansion of activity, with research slated to resume first in a modulated form. The process will be gradual and informed by public health guidelines and state, county, and local orders. We will base our decisions on data and do all we can to mitigate risk. The reality is that we will not be able to assemble as a full community for a good time yet, which is why today is so important. The commitment to discovery, to creating knowledge for the ages, and to improving people's lives today continues unabated, and we celebrate with you this shared mission. Thank you for joining us.
Thank you very much, Dr. Rosenbaum. I appreciate it. And to quote the Queen of England, who quoted a 1940s popular song, "We will meet again." I'm really honored to be able to introduce to you Dr. Davidow. He is a Caltech trustee, and he's a distinguished alumnus. He's director of the Aaron Diamond AIDS Research Center, the Clyde and Helen Wu Professor of Medicine at Columbia's Vagelos College of Physicians and Surgeons. He was recently profiled in The New Yorker, and in 1996, his discoveries about AIDS made him Time Magazine's Man of the Year. Now he's on the cutting edge of research on the COVID-19 virus, its causes, and the prospects for treatment. So Dr. Ho, thank you so much.
Well, thank you very much, Pat. And I would like to thank everyone at Caltech for the opportunity to participate in the seminar day. It's an honor for me. Thank you. Well, I'm going to speak to you about the SARS-CoV-2 pandemic, also known as the COVID-19 pandemic. And I'm speaking from the perspective of a virologist who has done research primarily on HIV, but to a lesser extent on SARS 17 years ago, and on influenza and a few other viruses. And of course, the SARS-CoV-2 pandemic has affected all of us and touched our lives and alter them for the foreseeable future. Everyone knows that this pandemic originated from the city of Wuhan in the province of Hubei within China. Shown here, it's central China, and it's the city is situated on the Yangtze River. And the manifestation of this infection was recognized in early December with a few cases shown here, and then gradually increasing in number in the city of Wuhan. This is from a publication in the New England Journal of Medicine describing the first 425 cases in that city. And as you could see, by the time we had our Christmas holiday here, there were already a number of cases of a strange pneumonia presenting to the hospitals there. And interestingly, many of them, shown in the darker shade of orange, had linkage to this seafood market called Huanan. And this is a picture, and of course that that market contained many live animals that were for sale. And because of the large number of linkages to the marketplace, the local CDC officials initially thought that this was due to animal to human transmission. The China CDC, the central CDC was asked to investigate at about the end of the year, and the seafood market was promptly closed. With the investigation, the team from China CDC was able to discover the cause of this outbreak as a novel coronavirus that was highly related to the SARS coronavirus from 17 years ago. And then upon isolation of the virus, the sequence of the virus was determined within a few days' time and shared with the rest of the world. And I should have mentioned that the WHO was notified at the beginning of January as well. And we all know since that point on, China has suffered a devastating outbreak concentrated primarily in the city of Wuhan in the province of Hubei. And that resulted in a lockdown in later part of January as this epidemic was taking off. But with that lockdown, China managed to control that epidemic down to a point where Wuhan reported 0 cases. I should know at this point that within China, even though there was this devastating outbreak resulting in about 80,000 cases, the rest of China
in all the major cities and other provinces, only less than 1,000 cases were reported. So the control of the outbreak was quite well done in rest of China. And then starting in late February, we knew that there is a outbreak in South Korea that was so small that it's hard to see on this graph. And very quickly thereafter, there was an outbreak in Italy. And we know that the Italian and Western European epidemics really took off in the month of March and persisted for a period of time. And of course, the US had the initial case actually way back in January with the traveler coming back from Wuhan. But the real epidemic took off in early March. And of course, we could see the numbers here shown up here, reaching over 30,000 cases per day in the US. And of the total that we now have worldwide, 4.5 million with 308,000 deaths. The US accounts for about 1/3 of the caseload and about 1/4 of the death. Now, just a little bit about the SARS coronavirus 2 and its genome here. This is the electron micrograph of the virus, and it's named coronavirus because it looks a little bit like the corona surrounding the sun as seen by astronomers. And it's these spikes sticking out are shown here. Schematically, there are also a number of other proteins on the surface of the virus particle, and its genomic organization is depicted here. I won't go into this in any detail, but suffice it to say that it's very much organized like other coronaviruses that are well known to all of us as virologists. And also in terms of the replication cycle, that's fairly well understood for coronaviruses. We know the virus will bind to a receptor on the cell surface and enter. And for SARS-CoV-2, the receptor is known as ACE2, which is a molecule that's important in regulating blood pressure. And it's on the surface of many cells, including cells in our airways and within the lung. And hence, the virus has affinity for our respiratory tract, but it could also infect many other organs. And once it enters the cell, via these vesicles, the virus will disassemble, and the genome is in the form of RNA, which could be copied by a polymerase, which will make more copies of the viral RNA to be packaged into progeny virus particles. But in addition, the RNA will make more messenger RNA, which will then be translated into proteins. And these proteins will assemble with the RNA genome into virus particles, which are then released. And it is expected that a single infected cell would make thousands and probably tens of thousands of progeny particles. We do know from the sequence of many SARS-CoV-2 now that it fits in with coronaviruses as shown in this phylogenetic tree or family tree. And shown here is SARS-CoV-2, and it is highly related to the SARS coronavirus that caused a worldwide outbreak in 2002, 2003. And that virus we know originated probably through the civet cat, which got infected by a bat species. But you could also see that there are other human coronaviruses shown in the burgundy and yellow color here. There's this MERS coronavirus, which caused an outbreak about 8 years ago, infecting a number of humans with a mortality rate of about 30%. And this virus, we also know, came from bat to camel to humans. And MERS stands for Middle Eastern Respiratory Syndrome. But down here are coronaviruses that have been infecting probably all of us for many, many years. And they cause pretty much a common cold or a flu-like illness and nothing more. But these three, SARS-CoV-2, SARS-CoV, and MERS, could cause severe disease. And it turns out that the closest relative of SARS-CoV-2 is a virus that was identified some years ago in bats in China, this rat G13. And it has about 96 to 97% identity with each other. And from the sequence information, we believe the SARS-CoV-2 is probably some sort of recombinant virus between this one and the SARS-CoV-like viruses. Now, a little bit about the clinical manifestation and key features of COVID-19. And this is probably something that you've been reading about or hearing about from the television. We now know that a lot of people who are infected by SARS-CoV-2 are asymptomatic. We know it's a large number. It could be anywhere between 20 to 40%, but the precise percentage is still not clear because we're not doing extensive testing of the population. But for a number of people who are infected, they will become symptomatic. And fortunately, a large majority would only have a mild manifestation of the disease, including primarily fever, cough, little bit of muscle aches, flu-like illness, essentially. You probably have heard quite a bit that a number of individuals will lose their sense of smell or taste. The exact mechanism for that is still not known. This virus is not known to invade neurons. But approximately 10% of the infected individuals will have a significant illness that may require hospitalization. But even more importantly are the 5% or so of the infected individuals who not only require hospitalization, but ICU care with need for oxygenation and intubation at times to be supported on ventilators. And the mortality rate is generally said to be 1 to a couple percentage. But if you look at the official numbers from Italy or the US, the mortality rate is about 6 to 7%. But that is because we're not testing a lot of people and not picking up a lot of mild or asymptomatic cases. So we believe the true mortality rate is around 1 to 2%. But if you follow the official figures, they're typically higher. The incubation period, that is from being exposed to development of symptoms, is said to be 3 to 6 days. Some cases may be a little longer, but this will cover about 90% of the cases. You may have been reading about what's called the reproductive ratio, and that is the number of one infected index case infecting X number of people. And that number is generally said to be 2 to 6 based on measurements, with a mean of about 3. So that means one infected person typically will transmit to 3 others during the growth phase of the epidemic. And of course, as you implement mitigation measures, that number will decline, and the caseload will decrease once the reproductive ratio drops below 1. There is something called the serial interval, or the generation time. That is, if the reproductive ratio is 3, that index case will infect 3 people over the serial interval of 3 to 6 days, or on average is actually thought to be about 4 to 5 days. So that's essentially the generation time for each successive wave of transmission. And then we now know that the virus could persist in an infected person typically for 2 weeks, but sometimes up to 3 weeks following the onset of symptoms. This is taken from a study that looked at throat and nasopharyngeal swabs and look at the amount of virus on the x-axis and on the y-axis, and on the x-axis days after the onset of symptoms. And you could see by 3 weeks or so, much of the virus is no longer detectable, but early on, there is a lot of virus, even as early as day 2. So this is probably the feature that allows this virus to spread so efficiently because of the high viral load during the early period of infection. And we believe this to be the case in asymptomatic individuals as well. Now, the clinical manifestation of severe COVID-19. Initially, we thought it was primarily pneumonia. As shown in this CT scan of the lungs, you could see consolidation of the lungs on both sides. There's no doubt there's pneumonia, there's respiratory distress, require ventilator support in severe cases. But our experience over the last 2 months, in particular in the US, has taught us that COVID is much more than pneumonia, severe COVID. It is essentially a multi-organ failure, affecting not only the lungs, but kidney. About half of the ICU patients require dialysis. We know there's a lot of cardiac abnormality, including heart failure. We know in many patients there's hepatitis and so on. The exact mechanism of how this multi-organ disease comes about is not very clear. We also see that there's hyperactivation of coagulations. You may have been reading quite a bit about strokes in younger patients due to blood clots. We know many of the lesions in the lung are due to blood clots within the pulmonary system. And then complement is part of our innate immune system, and that seems to be hyperactivated, and maybe these processes partially account for the multi-organ failure. And then over the last couple of weeks, there's been a lot of description of what's now called the pediatric multi-system inflammatory syndrome. In New York City, there are now about 100 kids who have been described to have this syndrome. It is basically high fevers, rash, inflammation of the major arteries, and some elements of what we know as toxic shock syndrome. And this initially was thought to be like a disease in kids post-viral infection called Kawasaki's disease. But now we believe it's both a combination of Kawasaki and toxic shock. The exact mechanism of this syndrome in children is also not clear, but it appears to be a post-infectious immune process that is not yet understood. Now, to me, the most impressive happening is the massive mobilization of science to address this pandemic, to develop drugs, to develop antibodies, vaccines, and to develop tests. And I've had, in my 30 plus years of research, I've never witnessed such a mobilization of scientists to address a single problem. You could say every large pharmaceutical company is trying to develop drugs right now. I would say there are 36 groups that are trying to pull out powerful antibodies from convalescent patients. And there are at least 4 or 5 dozen companies that are developing vaccines on a very fast track. And the testing issue, which I think we all been reading about and hearing about, I will not describe, but there are significant efforts underway to develop tests that would help us manage this pandemic. So coming back to drugs, we really don't have anything all that useful. The drug that was touted by our president has turned out to be non-effective based on a publication in the New England Journal only a few days ago. But you probably heard Dr. Fauci and other people talk about a drug called remdesivir. And that remdesivir is, whoops, it didn't show up, is a drug that blocks the polymerase. But it is only mildly beneficial. It would shorten the hospital stay of severe cases of COVID by a few days. So it's reminiscent of AZT back in 1987 for HIV/AIDS. It helped a little bit, but it did not make a major impact. And that's how we see remdesivir today. But it is a very important first step. In terms of antibodies, I will give you our example. And in terms of vaccines, you probably have heard there are several US vaccine companies as well as several Chinese vaccine companies that have already launched clinical trials back in April. And some expect to advance to phase 2 trial by summertime and perhaps would have some results by late in the year. But realistically speaking, this is a process that's going to take many months, not just a few months. I think if we had anything by early next year, that would be record setting already. Most likely it would require a year to a year and a half to have a vaccine completely tested and hopefully shown to be efficacious. So this is just the structure of the RP or the polymerase of SARS-CoV-2. It functions to copy RNA to RNA so that the virus could make infectious progeny. And this is a major target for drug development because for HIV and hepatitis C, there are at least 20 drugs that target the polymerase. And so there's a wealth of knowledge and a very rich chemical library from which to screen for compounds. And remdesivir falls into that category, although it was developed as a polymerase inhibitor for Ebola virus. Another major target for drug screening is protease, and the structure of which is shown here. This is, and the active site of the protease is shown here and here. It's a dimeric molecule. And protease is also another important target because of the drug development efforts that have taken place for HIV and hepatitis C. And we know quite well if one could intercalate a small molecule into the active site of the enzyme, you could block the activity of this enzyme and shut the virus replication down. And the enzyme is essentially a chemical scissor that would cut the large proteins of the virus into the smaller proper components. And if you gum up that chemical scissor, the protein processing for the virus would be perturbed and leading to production of non-infectious viruses. And on the right, I would just tell you that we've been searching for inhibitors that would fit into this pocket, and we now have found 3 chemotypes that would fit into this pocket and block the activity of the enzyme as shown for these 3 examples. For those who are used to seeing this type of data, you'll see that the activity is at this level, sort of a little bit sub-micromolar, which is not very, very potent. So these type of chemotypes would need to be modified iteratively into compounds that would shift the curve way to the left. And this is going to take months of chemistry, rounds after rounds of chemistry to define optimal compounds that could block the enzyme very, very actively. And so drug development effort is not going to proceed very rapidly. This is what we've done, but I would say there would be many dozens of pharmaceutical companies that are pursuing something very similar. And of course, they have many more chemists than we academic groups have. And so it's quite likely that there will be success on this front emerging from industry. Now, I mentioned earlier that there are many groups that are trying to pull out antibodies that would bind to the spike of the virus and neutralize the virus. And this is something that my team has also been working on. But we're not alone. There are already a number of teams that have published on coming up with antibodies that would bind this spike and neutralize the virus. But I would just on the right tell you about 40 patients that we have been studying to look at their serum neutralizing activity against SARS-CoV-2 in the laboratory. And on the top are patients from the ICU with severe disease. On the bottom are individuals with mild or non-severe disease. And you could see the curves here shift to the left compared to the curves down here. This means that, in fact, the ICU patients actually mount a more robust antibody response against the spike. And from some of the best cases here, we then take their blood and isolate their cells and go through a sorting process where we use the spike protein as the bait to fish out the antibodies, to fish out the cells, the B cells that are producing antibodies that will recognize the spike. And we have done this now for about, from 8 individuals, coming up with over 500 antibodies. And these have been systematically characterized to find ones that can very powerfully neutralize the SARS virus. This is the percent of neutralization. And as you crank up the antibody concentration in the laboratory, you neutralize more and more fraction of the virus. And I would say these antibodies down here are very close to ones that are being reported, but we have two antibodies that are quite special. You could see this one is neutralizing at 1 nanogram per ml. This is extremely powerful and is directed to this portion of the spike. And this is also very good, but not directed to this portion of the spike. It's directed to a different element. And so these antibodies that we're developing and others are developing in the field potentially could be used as therapeutic or as prophylactics. So for example, one could give a shot of the antibody to people who are high risk or who have been exposed, but as yet uninfected, to see if infection could be prevented. Alternatively, these antibodies could be administered as therapy, particularly early on in the course of infection. So I would say we're going to see clinical testing involving antibodies like these starting probably in a month or two, and we would probably have a dozen such antibodies in clinical trials by the summertime. Now, let me come back to more issues that would touch everyone. I've shown this slide already on the top, showing the China epidemic followed by the Western European epidemic and of course the US epidemic. But all of these are on the downward trend, but in other places, they're just beginning to experience the exponential growth that we went through during this period. And this includes Russia, which now has placed second only to the US in total number of cases, as well as Brazil. You can see the daily caseload is over 10,000. And India is on the rise as well. And given the large population at high risk there, it's very, very concerning. And it suggests that we're just going to have wave after wave of outbreaks spanning the globe, essentially. And even if we manage to control very well, we're at risk of spread from places that are hard hit at the time. And in particular, Brazil is in the southern hemisphere. As conditions improve with warmer weather and more sunlight in the northern hemisphere, the situation for the southern hemisphere is going to deteriorate. So what's happening in Brazil is going to be quite threatening. So there's a very good chance that this pandemic will persist and become seasonal. Very much like influenza that will bounce from the northern to the southern hemisphere as the weather changes, and then bounces right back as the weather reverses. Now, in terms of what's happening in our country here, you could see the US epidemic is on the decline overall, but that is largely due to the decline in New York. New York is obviously the epicenter of this epidemic in the US and was hit hardest, but its decline is accounting for much of the decline in the US. That simply says that the rest of the US is actually pretty steady. If you look at New York here versus some other states, Illinois is still on the rise. California is pretty much flat. So is Texas. So is Florida. There's subtle decline in others. But most of these, if the curve is close to being horizontal, that means the reproductive ratio is 1. and if you look at the absolute numbers, they range from about 500 to over 2,000 per day. And I think the leaders in Asia would think it's absolutely crazy to try to reopen under such circumstances. However, many states within the US are doing exactly that. So let me end by going back to the beginning. And first, the beginning of the New York City epidemic. This is the New York City epidemic with the first case recognized, first case of the community transmission recognized on March 3rd, and then low grade, and then it took off. This is the caseload within our medical center at Columbia. So it's accounts for about 1/20th of the New York City caseload. And as this was happening, we started our research on coronaviruses in February. And we thought, the epidemic was sweeping through China, a little bit of Korea and Italy. We thought maybe we ought to start collecting samples that were being tossed out by the clinical laboratory. So we started to pick up serum samples here. And we then later on tested these serum samples to see if they have any antibodies to SARS-CoV-2. And during that period here, prior to the first case within New York City, we found that about 3 cases out of 72, or about 4 to 5% of the cases were positive. And on the right is just showing that we did additional tests to confirm that those 3 cases are indeed antibody positive for SARS-CoV-2. So there, we documented 3 cases, and those 3 cases are shown here. But we know by the time you have detectable antibodies, the infection probably took place sometime earlier. We know that it takes 7 to 21 days to develop antibodies. And so we march back 10 days as a mean. And then there's an incubation period before onset of symptoms. And so that's another 5 days or so. So these 3 cases were probably infected around this time frame. And they represented 4% of the emergency room population. And so we calculated that there were about 48 cases that were missed. And then if you take this back by serial interval and reproductive ratio, we found that these cases, the New York introduction probably took place around here in late January. So the virus spread silently for about 4 to 5 weeks before the first case was detected. And then if we take this number and march it forward based on what we know of the serial interval and a more conservative reproductive ratio, we get almost 4,000 people within our catchment area by the time that first case was recognized. And if you multiply that by 20-fold here based on our catchment versus the entire city, there were tens of thousands of cases. So this points out the importance of surveillance, which was not being done. And that is because the testing was faulty at the time due to a mistake in reagent at the CDC. And CDC then provided guidance that testing would only be done in folks returning from other countries or contacts of travelers. And that is a critical mistake. Let me end by going back to China. We know that the SARS-CoV-1 virus originated from this particular horseshoe bat within China. The the bat species that carries the bat virus that's closest to SARS is slightly different, but it's also another horseshoe bat. And based on the viral sequences, we believe that the virus that's now causing our pandemic is some sort of recombinant between this virus and this virus, probably taking place 15, 20 years ago. So this is not based on the molecular clock that we know from the mutation rate of the virus. So this is not likely to be an engineered virus and then released intentionally or unintentionally from the laboratory. But why did, if that species gave us SARS-CoV-2, why Wuhan? And if you look at published literature, Chinese scientists have been, since the SARS epidemic of 2003, have been surveying bat caves throughout much of China. And the two species that are accounting for SARS and for SARS-CoV-2 are found in southwest China in the bat caves. And in fact, if you do testing of people in that region who live near the bat caves, some of them, a small percentage of them, show antibodies directed to the bat viruses. So bat to human transmission occur in such a setting. But Wuhan is right here, and it's a major metropolis. It's not known to have lots of bats. So why did this begin in Wuhan? And this is the question that many are asking, and there's no clear answer. I think I just said most of the scientists do not believe this is an engineered virus or intentionally released from the laboratory. But we need to come up with a proper explanation. And earlier I mentioned that the Huanan marketplace was tied to the outbreak. But there were already cases described prior to the cases linked to the marketplace. So it may not have been the origin, but it could have been the amplifier of the infection. So if we look at Wuhan, this is the Yangtze River coursing through the heart of the city. The marketplace is here. The Institute of Virology that's done much of the survey is located here, and associated with the Institute of Virology in Wuhan is the Chinese National BSL-4, the high safety containment facility that would do this kind of work. It's located here. The Wuhan CDC has a unit that's right next to the Huanan marketplace. And that unit, as well as the Institute of Virology, have been doing the bat surveys. So there's a lot of questions to be answered. Are the animals in the marketplace, they were tested? What are the results? Is there intermediary hosts? And of course, there's concern that folks who have been doing these bat cave explorations, could they have been infected and be the vector? And answers such as those have not been properly addressed. And this is why our National Security Council and those in the White House are making accusations. But I think it is also incumbent on the Chinese health authorities to share some of those findings with the rest of the world, because how a pandemic began is obviously crucial for us in avoiding future outbreaks. So let me end my remarks there. And I once again thank Caltech for the opportunity to present at Seminar Day.
Dr. Ho, thank you so much. And we have, as you would expect, a ton of questions lined up for you. One from Tony, PhD in 79. What's the best estimate of the percentage of people who die with coronavirus infections not having a pre-existing medical condition?
Well, that percentage will vary according to age. I think I don't have the exact numbers in my head, but if one were over 80, that percentage is as high as 15–20%. If one were in the 60s or 70s, it's probably in the order of 10%. And then it gets lower and lower for the younger folks.
So we should always be cautious rather than giving an aggregate number to specify demographics?
That's exactly right. I think you have to look at age and also some of the underlying conditions.
So Lynn, class of 83, wants to know about the possibility of reinfection. This is of great concern, especially when you talk about the prospect of 20 to 40% of people who perhaps were infected without knowing it, who think, well, gee, maybe now I have antibodies. Maybe now I'm immune from reinfection.
Formally, we don't have an answer to that question, and it's absolutely important. Does the development of the antibodies correspond to immunity? We obviously don't know that, but based on what we know of the common cold coronaviruses, having antibody is equivalent to having immunity. We don't know that for sure for SARS, and we don't know that for SARS-CoV-2. But time will tell. There have been anecdotal reports of people who tested positive, then negative, and then tested positive again. And that's been put forward as evidence for reinfection. However, that's probably more evidence for bad testing.
Joe wants to know what your level of confidence is that a vaccine will be found, given that we still don't have, for example, a vaccine for HIV.
A very good point. I think we're moving in record speed to come up with vaccines. I mean, it's amazing that we have over close to a dozen vaccines into human trials already, and that's absolutely amazing. But there's no guarantee that they will succeed. There are reports in the SARS literature that suggest vaccines potentially could do more harm than good. So that's a worrisome concern.
How does that happen?
Specifically, the antibodies that are raised by a vaccine would bind the virus when it comes in, and then it would be taken up by certain cells in the immune system called monocyte macrophages, and then they somehow would induce disease. And that's been described not just in one paper, but multiple papers. So that's a concern. But still, most of the people feel that that successful vaccines could be developed for this one, contrary to the situation with HIV, because the HIV envelope protein on the surface is very, very complicated and very difficult to elicit good antibodies. This one, based on what we're doing and based on what others have reported, it is fairly easy to develop antibodies that would neutralize the SARS-CoV-2. So I'm on the optimistic side.
The push for a vaccine has been likened to the Manhattan Project that developed the hydrogen bomb. And the question is for some people whether countries will be on their own track to develop and not share vaccine information, or whether there will be, as you said, this global community of scientists.
Yeah, well, our president just announced Operation Warp Speed for the vaccine effort. And the Manhattan Project was a unified project where everybody worked together in concert. I'm not sure that is necessarily the case for the global vaccine research effort right now. And it's not likely to be all unified under the American leadership because we have opted to go alone on almost everything related to science, right? From climate change to even addressing the pandemic. So I'm not convinced the rest of the scientific community outside the US would just simply fall under our leadership here. That said, I think it's still great to have a concerted effort within the US to develop a vaccine.
Raif from the class of 91 wants to know whether you have anything to say on the herd immunity approach that Sweden is taking, whether it's applicable to the US at all.
To me, it's a bit cavalier. In order to have herd immunity protect much of your population, you're going to have to allow your population to be infected by a large percentage. And that could be as large as 50, 60% of your population infected. If so, you would have many, many casualties. And I don't understand why anyone would simply accept that.
Carol, I'm sorry, go ahead, Dr. Ho.
I think the better strategy would be to apply the mitigation measures that we're all taking and flatten the epidemic curve and buy time. And I believe science will deliver. Given the massive mobilization, science will come up with a solution. I don't know whether it's a drug or antibody or a vaccine, but I'm quite confident, given the massive mobilization, that will come through.
You can ask your question of Dr. Ho. Go to the Q&A box at the Q&A icon at the bottom of the screen and put your first name or your last name if you wish, your class year, and if you are a parent or an associate or still a student, put that information in there too, please. Let's see, another question from James, who got his PhD in '78. The trend line of US daily cases shows a clear periodicity of roughly 7 days. Do you think this is just a result of reporting after weekend closures, or could it be the result of the resonant coupling between weekly patterns of social behavior and the incubation period of the disease?
I don't know. The curves I show are 7-day running averages. So we did that because weekend testing goes down and therefore confirmed cases would go down. And then it would spike on the Monday. So we took 7-day averages, but I think the testing schedule has some effect on that. There may be additional effects, and I'm not qualified to comment on that on the epidemiological side.
Is it too soon to be able to calculate all those factors, which seem immensely complex?
Yes, I don't think we could do those type of calculations right now. Maybe the Caltech folks working on artificial intelligence could do that.
All right, you heard it. There's your next assignment. John, class of '71, says, why do some counties such as Los Angeles County and Hennepin County in Washington State have so many more deaths than other counties? Again, to a lot of factors.
Yeah, there are many factors. I think, you know, I now live and work in New York, so I could speak to New York better than I could for California or Washington. In New York, we see certain spots with high death mortality rates, and it's primarily in African Americans and Hispanic populations. It has to do with you know, having underlying conditions, and you know, it's part of the inequality in our society. And if you look at Upper East Side, Upper West Side of Manhattan, the death rate is low, the infection numbers are low. If you look at the Bronx or certain parts of Queens and Brooklyn, they're high, and demographics explain that.
Is that historically true of pandemics too, the distribution among poor communities versus rich communities?
I haven't studied that extensively, but I think that is true of influenza and probably of other pandemics. The one I know best, HIV/AIDS, well, that is true of the present day picture in the US.
Here's Jim, class of 81, who says, how is the folding at home distributed computing project helping in the fight against SARS-CoV-2?
Could you repeat that question?
It says, how is the folding at home distributed computing project, I'm not familiar with that, helping in the fight against SARS-CoV-2? People's computers are being used to find possible enzymes and proteins about this. And it's kind of a citizen science thing or self-crafting, self-contributing to a larger data trend.
Yeah, so there's a lot of in silico work, computing work, to find relationship between molecules and proteins of this virus, and that has produced some leads for scientists to chase down, in particular related to drug development. But I'm not aware of any major outcome from that effort at this point. It's still very early though.
I suppose, again, self-reporting and self-selecting doesn't give you the consistent databases that you would like if you were asking and looking on your own.
Right.
So, let's see. Carol asks, some researchers have indicated a link between vitamin D deficiency and the severity of infections. Can you comment on whether we should all be taking vitamin D supplements? It's like rickets again, right?
I haven't seen any scientific evidence that vitamin D supplementation would be helpful. But now most of us, or many of us, are indoors at home.
Like now.
Vitamin D would be advisable on that account.
Not because of COVID-19 though.
Not in my mind at this point.
Rahul from the class of 96 says, a recent paper from UCSD suggested that many people, perhaps as high as 50%, have existing immunity to COVID-19 as a result of their exposure to past human coronaviruses. Can you comment on that?
Yeah, I can't comment on that study in particular, but I could comment on the point. We have looked at hundreds of samples of serum stored in the pre-COVID era. And we don't find any that's positive.
I'm wondering, as we talk about the recurrence of this, whether we're looking at tuberculosis of the 21st century, which is an endemic disease. Some people are exposed to it, never come down with it. Some people are exposed to it, come down with it, and die. Is this, without a vaccine, is this the prospect that we might be looking at?
Well, that's what we're looking at currently. A lot of people are infected. A large majority do quite well. And some are even asymptomatic, but some succumb to the disease. Exactly why, we don't know. We do know that age and underlying diseases, hypertension, obesity, diabetes are all risk factors. But no doubt there are also genetic factors, and there's a race on to find those genetic factors. And if we do find them, they will be helpful in managing the pandemic, because we could try to protect, largely protect those who are at greater risk based on genetics.
We have John, who got his PhD in '83, asking whether you can comment on the racial, national, and socioeconomic differences in the impact of COVID-19. You've already touched on some. Is there anything else?
No, that's such a broad question. It's more social science, and as a basic scientist, I don't think I'm the one to address that.
Leon from the class of 10 says, how fast is the virus evolving, and have we seen evidence of novel strains?
Yes, like all RNA viruses, there's a high mutation rate, so it's evolving. this virus does have an enzyme that will fix mistakes, so it's not as error prone as other viruses. However, there's a rate, and we already seen lots of mutations, which have as yet not resulted in any functional significance. But we could trace, for example, based on the mutation, trace that most of the viruses in New York City were introduced from Europe, not from the West Coast, and not directly from China. So our lockdown, our shutdown of flights from China prevented direct infection from China, but we didn't do so for Europe, and that's how New York City got hit.
Anjul, who is a postdoc right now, wants to know whether you're optimistic about current funding trends in science for this issue. It may be short-term, a lot of interest, long-term, it's always the question.
Well, short-term, there's no question there's massive infusion of funding into life sciences, and in particular for anything related to the pandemic. Enormous amount of funding, and I know many institutions are positioning themselves to take advantage of that. But would that come at the cost of other scientific research? And that would be a concern for me, especially given an administration that has not paid a great deal of attention to science and at times ignores or trivializes science.
Audrey would like to know whether the reproduction number of 1 is the lowest achievable in the US with the current social distancing policies in place.
No, we cannot accept a reproductive ratio of 1. We have to drive it to close to 0. In fact, if you look at what Wuhan did, they drove it to 0 before opening up so that there were no cases. And South Korea drove it to very, very low numbers before opening up. And even when they did at such low numbers, you probably read in the past week, there's an outbreak, small outbreak. So I think opening up with thousands of cases per day is very, very risky, especially when the reproductive ratio is 1. That means when you open up, it's going to grow to a number larger than 1. And if it goes back to 2 or 3, you have the exponential growth that we have seen already. This is why Dr. Fauci is so cautious.
Let's take a last question from Evan from the class of '84, and it may be too soon to know this about the long-term effects on people who've had serious infections.
Yes, that's also a very, very interesting, important question. We are seeing now that pediatric syndrome is a post-viral complication. And that's fairly acute. That is, it follows the infection by a few weeks or a few months. But for SARS, we do know that substantial number of recovered patients develop a condition called pulmonary fibrosis. So the lung is injured and becomes fibrotic. And it's a major impairment of respiratory function. Would that happen here? I think we have to wait and see. But I think it's quite likely, given the extensive damage that's taking place within that organ.
Well, Dr. Ho, thank you for being here today, and thank you too for your work, which holds out so much promise to so many people. I know we're all grateful. Thank you so much.
Thank you, Pat, and thank you, Caltech.